Welcome everyone to our 5th GBM. This is the Society for Conservation Biology Florida chapter. And I'm sending a link in the chat to sign in. For the people in the room, I'll write the link on the board a little later, but yes. Oh, okay. Okay. Cool. So yes, thank you, Bruce. And yes, so this is our 5th meeting actually. So yes, I think everyone here knows who we are, but we SCB does conservation research, and we invited a conservation professional here today about Lepidoptera research, so moths and butterflies, and yeah, just to go over our chapter. We do bi-weekly GBMs, but we also have a GBM next week because spring break happened. So we're just doing 2 in a row for these 2 weeks. And then we also do various events and volunteering. And then for those who attend, we have our point system for you to be able to have merch and attend our end of year trip, and for future officer positions if you're interested. And then we also are working on putting TerraCycle boxes in New and Ziegler Hall for you to put these items, bottle caps, fabric, personal care containers, and office supplies for them to be recycled, because they can't usually be recycled through our waste stream. So keep an eye out for information on that. And then our upcoming events are a beach cleanup this Saturday with the Sustainable Ocean Alliance and Marine Bio Club. And it's on March 19th at 10 a.m. And yeah, so sign up for that on the information on the link in the chat. And we'll arrange carpooling after enough people sign up. We'll send out more information later in the week about carpooling. We also have a fundraiser on March 26th, which is the Saturday after that. And it's from 11 to 9, and it's on the Blaze on Archer Road. And then be sure to tell them you're there to support SCB, and we will be sending out a flyer for you to show them as well on your phone. But yes, all this information is also in the link that we sent. And then we have details about our end of year trip coming up in about a month. So we're going to the Moat Aquarium in Sarasota on April 15th and 16th. We'll be doing a breakfast with the sharks event that morning on Saturday the 16th, and then a day at the aquarium, and we'll be staying at an Airbnb that one night. And there are limited spots, so, and it's based on your active member status and how many points you have after that, because we can only bring 12 people. And so, yeah, we'll send out the itinerary to those who can go. And the sign up for that closes tonight, and it's also on the forum. What the heck? Okay. Oh, okay. So, yes, without further ado, I'm going to announce our speaker for tonight, Dr. Jaret Daniels. He's a curator at the McGuire Center for Lepidoptera and Biodiversity at the Florida Museum of Natural History.
Thank you. Thank you all very much. I really appreciate being here and for the invitation. And I'll go ahead with my presentation. Can all of you hear me okay? Is the audio good? Okay, wonderful. Well, thank you again for having me. As was mentioned, I'm an insect ecologist. I work at the Florida Museum of Natural History's McGuire Center for Lepidoptera and Biodiversity, and also in the Department of Entomology and Nematology. And while I do work broadly on insects, I'm going to talk tonight specifically about at-risk butterfly conservation efforts, which is probably one of the components of my work that I'm most fond of, and is probably the most rewarding element, just because we do get to work on a number of exceedingly rare species and look to develop new ways and new best practices to help ensure that we are making progress with their recovery. And so obviously, we all know that we're in this unprecedented state of existence on this planet with biodiversity loss, and there's been a lot of attention recently with insects, some undoubtedly overblown. Here are just some of a smattering of headlines and research article titles, but there's no doubt, obviously, that in particularly well-studied groups such as Lepidoptera, bumblebees, and some other groups, that there's clear evidence of significant declines at a global level. So this kind of provides the foundation for a lot of my work, because I became interested in insect conservation kind of long before it was really a kind of emerging field. I mean, it's been around for quite some time, but traditionally when you came down to do a degree 30 years ago in entomology, most folks were interested in controlling pests or dealing with insects in agriculture. And so trying to work across landscapes is really something that my lab does. So we work in a wide variety of systems, from wildlands to agro-ecosystems to rights of way, whether it be for utility providers or transportation providers. And we also work increasingly in the built environment, an area that has a lot of opportunity for recouping or recapturing resources and biodiversity, especially when it comes to insects. And this is ever so important because of those declines. But I will say that I really do love this, this graphic on the left-hand side of the slide. This was from a paper by Wagner et al. In 2021, and it really exemplifies sort of the threat to biodiversity globally. And this one is obviously directed particularly for insects, but outside of sort of the big kind of drivers of decline, habitat loss, fragmentation, and climate change, it really is death by 1,000 cuts of a number of kind of drivers kind of eating away at the margin that helps to destabilize systems and ultimately continue to cause species collapse at a global level. So in order to come up with solutions for this myriad of challenges, threats, we really have to look at all systems. No longer can we be relegated to say that wild lands or conservation lands are sufficient, because we're seeing declines on those conservation lands. So that means that all landscapes become increasingly important, and we have to try to develop ways in which to recoup resources and manage biodiversity across all these different landscapes, because collectively they're going to be essential to maintaining the functionality, stability, and productivity of our systems moving forward. And insects are a great model because they're small, they occupy all of these landscapes, and they're also, as you all know, really critical providers of ecosystem services, such as nutrient recycling, decomposition, plant pollination. So they are functionally quite critical for biodiversity at a global level. And just to show you that, you know, while I am particularly focused on Lepidoptera conservation, my lab does do a lot with a wide variety of different landscapes, and we publish predominantly in looking at developing best practices across these landscapes, whether it be to enhance crop pollination within Florida or globally, whether it be dealing with urban environments, individual managed species of insects such as bumblebees, or really charismatic taxa such as the monarch. We try to work across these with the kind of the united factor being the development of best practices for insect conservation, so that ultimately we can understand how to manage these systems more appropriately to ensure that diversity and richness are not lost through time. And if you talk specifically about insects, or butterflies, I should say, and within our home state of Florida, there's really been a pretty strong decline, especially in South Florida over the last 20, 25 years. And much of this has gone under the radar, so to speak. And so the three species that you see here, the three taxa, the Ammon blue, Rockland grass skipper, and Zesto skipper, two of these were declared extinct in 2016, and one, the Ammon blue, has not been reported in over a decade. So it's clear indication that we are losing diversity in our home state without very little knowledge public knowledge of this. So these are fairly charismatic species, but it's hard to even find a newspaper article or an online blog about these organisms declining, but it is happening quite rapidly within Florida. And so we have been working with a variety of different diverse stakeholders to try to address these declines, both in Florida and beyond. And I should also say that my lab is pretty unique in the fact that we have funders that are fairly diverse. And while a lot of labs may focus on individual funding streams such as National Science Foundation or other federal sources, my lab views funding for conservation kind of like a stock portfolio, where you have a very diversified list of funders, some major ones that dominate, but a wide variety of funders, private, individual donors, individual corporations, because those gets you through those lean years and enables you to kind of diversify your actions and your ultimate workload. So one unique funder that we have built a long-term relationship with is the Disney Conservation Fund. And most of you know Disney as a media company or an amusement company, but they have a long-standing history of 25 years of giving to conservation. But about 9 years ago, they decided that while we do give money to smaller projects, kind of capstone funding for projects globally, they really wanted to have a deep and impactful strategy moving forward. So they developed a new campaign called Saving Wildlife, where they would invest deeply in 10 organism groups globally for a 10-year period. So it's essentially unprecedented for most funders to have that kind of long-term viewpoint. They understand that conservation, it does not happen overnight, it takes time. And so fortunately, one of those organism groups were butterflies. And we, along with the Archie Carr Center, received 2 of these awards. We're the only university globally to receive 2 university awards, one dedicated to butterflies, one dedicated to sea turtles. And so as part of this work, we tried to think as broadly as we could, and we focused predominantly on 2 kind of locations where both endemism and diversity were high and species loss was rapidly accelerating. And these turned out to be Florida and California, not because that's where Disney does business, but these really harbor the majority of the listed Lepidoptera in the US, and they harbor some really unique key taxa for conservation. So at the end of the day, we drilled down to focus on 42 different taxa in Florida and California. You can see some of the diversity of species here, including the ever-present monarch butterfly highlighted in the second column. So this includes declining taxa, as well as those that are federally listed as endangered or threatened under the ESA. And as part of this work, we do a lot of conservation planning with partners, and we also try to publish, like we do broadly with insect conservation, on developing better practices for recovery and conservation of Lepidoptera as a whole, so that we can learn from these models, we can develop new platforms, we can move the field forward in a reliable way so that we are actually making impact in the long term. And for much of our Disney work, we employ the conservation measures open standards for the practice of conservation. And this is an iterative process with a adaptive management cycle ultimately, so that we can kind of carve out the direction we want to go. We have clear metrics of success, and we can intervene sooner than later if things are not going the right direction, because the last thing we want to do, of course, is go down a 10-year cycle and at the end of the day come out with not the result that we were hoping for. And so this allows us kind of in the moment adaptation to understand where we're going, understand and appreciate and incorporate our stakeholder needs and desires, and also react to current research and what the organism itself is doing. So we try to plan things out as well as possible. And we use the online software called Marathi, which develops kind of decision trees and results chains so that we can easily communicate to stakeholders what we're doing. And we can react in the moment if things go a different direction than expected, so that we can adapt quite readily and quickly to the moment, ensuring that we put our best foot forward for the ultimate end goal of recovery. And as part of this effort, we involve our stakeholders quite a bit, both at the beginning of projects to ultimately understand what their needs, what their desires are, what potential limitations they have. We also drill down into what is known about the organism itself. And then where possible, we also do this in the field so that we try to ensure that across any conservation project, we work on the ground with our conservation partners. We do training, we work with land managers, in the case of, say, prescribed fire. We work with other stakeholders, private as well as agency stakeholders, to understand how to move projects forward more effectively, so that we develop an equally transparent and equitable platform to deal with species. Because as you know, many of these species, depending on where they're located and the lands upon which they exist, there's some really complex challenges that might involve a wide variety of private stakeholders, tribes, management, a whole wide variety of issues that need to ultimately be addressed and mitigated. And I'm going to take the next a little bit of time to kind of talk more specifically about a few individual taxa, just to give you an idea of kind of the depth and breadth of the work that we do. And so starting out with this little guy, this is the Miami blue. This is a small polyomatine blue. The genus is found throughout the Caribbean, but this is a Florida endemic. It's found historically in coastal areas of Florida from about Merritt Island south and around the coast of Florida to the Tampa-St. Pete area. But over the last 30 years or so, it declined rapidly north to south to the point where it was thought to be extinct in 1999. It was rediscovered in a small state park in the lower Florida Keys. And today it exists in only a few remaining fragments of land to the west of Key West, namely the archipelago of the Marquesas Keys and one small island called Boca Grande. And these are wonderful environments in which to work, accessible only by boat. So here are a few of my technicians, Sarah Steele Cabrera and Taylor Hunt, going out to the boat, moving between island to monitor for this butterfly. And this is kind of the coastal strand habitat that this butterfly occupies. It feeds on two fabaceous hosts, Florida Keys blackbead and gray knickerbean. And all of these islands are only a few feet above sea level. So obviously future climate change is a major concern, as is the intensity and frequency of tropical cyclones. And it also has a really unique relationship with several native and non-native ant species. In this picture, you see two late instar larvae and two worker Florida carpenter ants actively tending the larvae. And this is a fairly common symbiotic relationship or mutualism in the family Lycaenidae. It's much more well-developed in the old world than the new world, but it is an important factor nonetheless for the long-term survival of this butterfly. And just want to show you a quick video. So hopefully you can all see this. So this is a male carpenter ant, worker carpenter ant, and he's coming across a Miami blue larvae, and you can see the tentacular organs kind of pulsating out from the rear of the caterpillar. And so each caterpillar has dorsal nectary organs, which release kind of a carbohydrate-rich solution, which the ants use for food. And then those kind of bottle brush organs that are popping out emit a variety of different semiochemicals that appease and modulate the ant's behavior, so that it tells the ants that I'm a friend, don't eat, I'm not going to cause you harm. But they can also kind of invert those tentacular organs and call the ants over to their defense if they are being attacked by another invertebrate predator, such as a wasp, another ant species, whatever the case may be. So this really mediates the response and the interaction between the larva and the ant. And this is incredibly important ultimately for the survival of this taxon in Florida, because if you look at the role that these ants play when larvae are being tended versus when there are no ants present, that graph on the right-hand side shows that when ants are absent, almost 99 to 95% of the larvae are predated by invertebrate predators. But when the ants are present, they're wonderfully successful at thwarting the attack of any invertebrate predator, because these are fairly large, robust ants. They have large mandibles, and they can be quite numerous around the individual larvae themselves. So they are a very effective defensive mechanism for those larvae. And of course, the ants also benefit from the larval association by increased food quality and quantity, which adds to or leads to larger brood size in the ants themselves. So this is a really great mutualism that is truly mutually beneficial to both organisms. But as you can see, in areas where potentially invasive species might disrupt this relationship, the Miami blue might have a really hard time surviving in the future. So this role is really important. And then for this butterfly, we really consider this in many ways a model system, because through this effort, we really want to try to test and develop robust best practices to design ultimately reintroductions for this butterfly. And if you look at kind of the field of reintroduction biology, historically, a lot of this was driven by sort of case studies of releasing organisms and ultimately determining what happened after release. But invariably, much of this work was not done by asking questions in an a priori method. And that is changing quite rapidly within reintroduction biology, but it still has a long way to go. So our goal with this effort is to really look at how do we ultimately maximize establishment of these populations in the wild, and how do we ensure persistence long-term. So we wanted to do so by testing a number of a priori hypotheses that ultimately dealt with what we were releasing in the wild. And so as part of this effort, we developed a fairly large captive breeding population based here at the Florida Museum of Natural History. This is a continuously brooded butterfly, so we can produce about 1,200 organisms per month when this colony is going quite well. So that's a lot of organisms for a highly imperiled taxon to deal with. And that gives us a lot of rich numerical breadth to ask these questions with, if you will. And so starting in 2019, we looked at a few locations within the historical range of this butterfly as potential reintroduction sites. And these include Long Key State Park and Bahia Honda State Park, which is the last remaining extant population historically within the island chain of the Florida Keys, outside of the current extant range of the butterfly. And key to our effort with this was looking at questions that relate to the life stage being released in the wild, the quantity of organisms being released, and ultimately whether these were soft or hard releases, essentially whether they had some level of predator exclusion, and potentially that would help with establishment in the wild. So in this picture, two of my technicians, Matt Standridge and Kristen Rossetti, are setting up these little PVC kind of vials, if you will, and inside each of those is a little screen at the bottom and one or two Miami blue pupae. And then these can be actively deployed in the field. They can be GPSed, and we can go back in time after release and look whether those larvae successfully eclosed as adult butterflies, whether they died under natural causes, or whether they were predated. And so ultimately our goal with this was to test not only the life stage being released, but the method of release itself. So through this effort, we tried to test a number of different release containers that were deployed on the ground, in vegetation, hanging from vegetation, had different quantities of pupae in them for ultimately being released, and go back and kind of evaluate the success or failure of these various methods. And then we also looked at releasing larvae in the field as well as adult butterflies in the field to truly test how impactful or how important is life stage at release for ultimately establishing populations in the wild. And beyond this, we also deployed large pop-up cages in the field. This is my technician, Taylor Hunt, based out of the lower Florida Keys, deploying several walk-in cages at Long Key State Park. And inside here, we would have host material, nectar material, and then some variation of either larvae or either pupae or adult butterflies to test again how important was predator exclusion, and does life stage at release, even in this context, does that really matter, and how important is that in ultimately establishing a population within the confines of this cage in the wild. So in this context, we use an alternating method of using adult butterflies and pupae varying by month at 2 different locations in the field, and then collected a variety of in-cage measurements to kind of quantify more effectively what variables were going to go into our models that we could use to look at best fit for what was driving the ultimate success of these projects. So again, looking at everything from host plant species and cover to bare ground leaf litter. We did active adult counts, larval counts, pupal counts, egg counts. We measured everything from precipitation and temperature and looked at organism survival, and ultimately trying to kind of dial in on what were the driving variables that led to ultimate success of establishment in this context. And then beyond kind of work like this, we also try to do a lot on the recipient site issue as well, because this butterfly, again, was widely distributed across Florida. And unlike many other taxa, habitat loss was not a key driver for its decline. There still is essentially a lot of potentially suitable habitat out there. But when we're starting to look at where we put this butterfly back, we really wanted to have a robust metric to evaluate site per site conditions so that we could ultimately make the best choices and work with our stakeholders to identify what we believe to be the most effective sites to target for organism reintroduction. So we work with Fairchild Tropical Botanical Gardens, and we developed a long list of potential criteria, biological and logistical criteria. We went to these habitats, we mapped out the habitat at each location, individual host plants and coverage of host plants, nectar plants, structure within the landscape. And then we also measured it against a number of logistical criteria that often get overlooked a little bit when it comes to conservation. And this might be something as basic as just willingness of the land manager to receive the organism to manage appropriately for that organism. How accessible were these sites, or how easy were these sites for regular monitoring? Did you need to take a boat out to them? Were they accessible by vehicle? Did you need to hike in a long distance? How much public disturbance or accessibility do these sites have? And was there some buffer from mosquito control abatement within Florida? So all of this went into a larger matrix that allowed us to effectively drill down and identify high priority sites. So we do try to hit it on both sides, both the organism reintroduction side and the site-specific variables, to ultimately ensure that our projects are as well thought out and successful as possible. And then jumping to another Florida butterfly. So this is Schaus's swallowtail. This is, by contrast, a very large and showy butterfly. It's the only federally listed swallowtail within the United States. And this butterfly is limited entirely to conservation land areas in Southeast Florida. It currently is found in only 5 locations: Crocodile Lake National Wildlife Refuge, Biscayne National Park, Dagny Johnson Key Largo Hammock Botanical State Park, John Pennekamp Coral Reef State Park, and right here at the Florida Museum of Natural History under culture at the museum. It's exclusively limited to tropical hardwood hammocks. It feeds on only a few retaceous host plants. It has 1 to 2 flight seasons per year in the spring, and then a later, smaller flight in late summer, early fall. And much of the work that we do with this butterfly deals with just monitoring population numbers from year to year. And we do this a number of ways: by visual counts. We also do mark recapture. We do distance sampling. It really depends on the location and the complexity of the individual site that we're working in. But as many of you know, if you work in Florida, and particularly work in areas where they do not spray for mosquitoes, these can be pretty challenging environments where you have to be dressed in full mosquito gear. Otherwise, you get slaughtered by the really millions of salt marsh mosquitoes. And in many of these locations, like this site here in Biscayne National Park, of course, there is no mosquito control abatement. And historically, this butterfly has a very violent history. You go back to 1986, a little after when it was listed at the federal level, you can see the numbers kind of going up and down, and the kind of key marks on this graph, this kind of rudimentary graph, are highlighted by the star in 1992. That's when Hurricane Andrew struck Southeast Florida and made a direct impact to the remaining extant populations of this butterfly. And at that time, it was thought that indeed Andrew had made a death blow to this butterfly, that there would not be any chance of survival. Fortunately, they did actually survive the storm, and fortuitously, we also had collected prior to Andrew a nucleus of eggs to take back to the Florida Museum that served as the starter stock for our captive breeding program early in the 1990s. And you can see recovery after that, and then in around 2000, 2001, all of our federal funding dried up. And from about 2004 to 2011, there was very poor monitoring and very much anecdotal understanding that this butterfly still wasn't doing particularly well. And so in 2012, we enacted, as part of the Imperiled Butterflies Working Group of Florida, a group of state and federal agencies, the first comprehensive range-wide survey for this butterfly, which deployed about 100 people on the field, about 12 different Florida agencies and organizations, and comprehensively tracked this butterfly for the entirety of the 2012 flight season. And at the end of that season, the numbers were woefully low. Only 4 individuals were recorded in the wild. So at this point, we were very much worried, were we watching an extinction event happen within Florida? Fortuitously, we did capture a number of organisms that we were able to take back to the Florida Museum of Natural History at this time. That started a robust captive breeding population, which still serves today as sort of the stock for organism reintroductions. And luckily, these are butterflies that are fairly easy to breed in captivity. But as you can imagine, if any of you have raised insects in the past, you know that a lot of work goes into taking an organism from egg to adult. And even within insects, although they are small, one individual full-grown caterpillar of Shasta swallowtail can defoliate a 3-gallon wild lime tree during the course of its development. So if we can produce 1,000 organisms in a growing season, if you do the math, that's over 1,000 plants that we also need to maintain. So it's a lot of work that goes into the back end of this type of programmatic ex situ work. And then based on how productive the captive population is, we use organisms for release into the wild. And this is Matthew Standridge releasing a second instar larva within Biscayne National Park. We do a variety of life stage releases, much like we do with the Miami Blue, to test the efficacy of life stage at establishment. And we also try to work with our stakeholders and involve our stakeholders directly in this on the ground work. So in this slide, as an example, you can see individuals working alongside me that are from U.S. Fish and Wildlife Service, the Florida Fish and Wildlife Conservation Commission, and the National Park Service. And these individuals are ultimately responsible for their conservation. But as you might imagine, that many of these folks don't get into the field very often. And in some cases, they might be the first time that these individuals have actually seen this organism in the wild. So it's very rewarding to involve our stakeholders directly in the on-the-ground conservation work themselves. And unlike many other highly endangered organisms, we also have a cadre of community scientists working alongside us. And they've really been on the ground with us from the start of this project. And they were actually part of a publication looking at how citizen scientists or community scientists can ultimately develop and build a statewide at-risk butterfly database. And so in this context, they were actively on the ground and throughout northern Key Largo in very challenging conditions, monitoring for this butterfly. And we rely on them for a lot of real-time data that feeds into our understanding of how this butterfly is doing. And I'm really happy to say that since that 2012 low point of 4 individuals, we've come a long way. And today we've recorded really this past year was the most productive year in memory, with almost 1,700 individuals found in the wild. And this exceeds even some of the highest numbers from the mid-1990s. So we are certainly seeing the results of the actions of many, many individuals helping on the recovery of this butterfly and moving the needle in the right direction to hopefully have this butterfly secure and around for the foreseeable future. As part of this effort, we also are trying to establish it in several new locations within the historic range where it currently does not exist. As part of the currently approved federal recovery plan, it calls for the establishment of 6 new self-sustaining populations. So we started in 2020 in the middle of the pandemic to release individuals within Lignum Vitae Botanical State Park in the lower Middle Keys. And the slide that you see here are a group of stakeholders, including myself, releasing individuals on that island. Both adults, larvae, and pupae were released. And from the work that we've done, I'm happy to say that in 2021, we started seeing the fruits of this work as well, with the first individuals being recorded within Lignum Vitae Key really since the 1940s, and successful evidence that many of the organisms or the life stages that we put in the wild survive to become adult butterflies. So we have a long way to go, but we are indeed making progress. And then beyond, the work on super rare species, we also do a lot of work on declining taxa. And this kind of feeds into what I mentioned at the beginning of the talk, where we try to look at increasing landscapes within the built environment for how they can be important for insect conservation. And of course, the monarch is kind of the, idyllic, iconic insect. It's the most well-known butterfly in North America. It's probably the most well-known insect in North America. It's really energizes the public when it comes to conservation. It really has kind of turned the tide with pollinator conservation between agencies and the general public. So it's a great model organism to kind of move forward with conservation work. And so we've tried to work on it as much as possible, and particularly within the Deep South, because a lot of the work has been done in many other areas of the US, but the Southeast and Florida in particular didn't historically have a lot of research on the monarch. So we do everything from long-term population monitoring to the importance of different landscapes within suburban environments or roadsides for understanding how we can ultimately work together for monarch conservation. And all this kind of feeds into work with other partners, including the Association of Zoos and Aquariums and their initiative called SAFE, which is Saving Animals from Extinction. And they have an individual monarch SAFE initiative that launched 2 years ago to fight the decline in the North American monarch. And we also work with transportation and energy providers on the monarch nationwide candidate conservation agreement with assurances to protect and restore landscapes within those industries for the monarch itself. And as part of that work, we also mapped out roadways in North Central Florida, monitoring milkweed populations. And so in this context, the map that you see on this slide, the roadways highlighted in blue have high density stands of milkweed species for the monarch. And so if you're a DOT and you are trying to manage your roadsides effectively for the monarch and ensure that these populations are mowed at the right time of year, they don't become sink populations over time, these might be locations that you might wish to target. The roadsides highlighted in red have ultra high densities of milkweed populations. And such populations are increasingly important for the monarch because we started to see declines in milkweed on many conservation land areas. And as you can quite imagine, that roadsides actually harbor fairly high density stands and diversity of milkweed species across Florida. So these landscapes are ever more important now than they have been for insect conservation, and particularly for the monarch in this context. And we're also launching a new project with the Department of Transportation using drones and AI to monitor milkweed populations through time. Since we have a fairly robust initial baseline of data, we want to understand how FTOT's management long-term is affecting these milkweed populations. Are populations remaining stable? Are they declining? Or they're increasing? And trying to use tools like remote sensing, AI, and drones to effectively reduce labor costs and really enhance and build models that can be applied to other landscapes, I think has a lot of utility. So this is a project that we're launching at the end of this month for 2022 in collaboration with several private landowners and the Department of Transportation. And then we're also working with the Department of Transportation on other DOT managed lands, including retention basins. If you look along roadsides, it's not just the road verge that FDOT maintains, but they maintain dry and wet basins for stormwater management and runoff. And these are totally off the conservation vernacular, but these are highly appropriate locations to revegetate or restore for pollinator conservation or for monarch conservation. So we have a 3-year pilot looking at restoring these systems with a variety of pollen and nectar producing plants and multiple species of milkweed in support of pollinating insects in general and the monarch. And so these are large active restoration and planting projects in areas where DOT can be a little bit more flexible on their management compared to roadsides themselves. And then along the same line, for moving into the built environment, we have a new project that was launched as part of an Opportunity Seed Fund project funded by the University of Florida to develop a UF wildlife-friendly plant certification program to understand that as a consumer, when you're buying plants for your home garden or for a municipality or a schoolyard garden, that you have some understanding of the kind of data-driven priorities that go into making sound choices. And so right now, if you go to a big box store and you buy a plant, there's really no metrics behind that plant that said it's good for butterflies, bees, birds, whatever the case. And there's also no metrics to understand that it was produced from the growers in an efficacious way to reduce impact to the target species you wish to attract. So again, we're using monarchs as kind of the model organism here and tropical milkweed as the model system, looking at both the evaluation of these plants for their attractiveness to wildlife, but also from the pesticide impacts, the inputs during production to mitigate the eventual impact to the butterflies themselves. Because right now, if you buy a plant that has been treated with a systemic insecticide, there's a good chance that 95 to almost 100% of the organisms using that plant in your landscape could suffer some lethal or sublethal effects from those pesticides. So producing them in an environmentally sustainable way that provides good pest efficacious control, but minimizes non-target impact is going to be critical to moving the green industry forward for wildlife conservation and making sound choices within urban and suburban landscapes. And then we also try to, where possible, develop tools for studying rare species. And so we have a number of projects right now looking at kind of range-wide population genetic structure. And as you might imagine, it's always really critical when you're dealing with incredibly rare or listed species that you minimize the harm to these organisms during genetic sampling. And so we piloted another project with this little butterfly called the frosted elfin that did not actually sample the butterfly itself, but used the hatched eggs as a way of collecting population level genetic diversity that could be used for range-wide assessments of population structure. And so we were able to actually get really sound results from only a few eggs sampled in the field over time to really drill down into really good understandings of kind of the range-wide population structure and genetic exchange across the landscape for this particular butterfly. And then I'll end a little bit by just talking about some of the outreach and education that we do, because this really goes hand in hand with the research and the best practice work that we try to accomplish. We are, of course, in the Florida Museum of Natural History, where we receive over 200,000 annual visitors. Public understanding of science is really critical. So we take public outreach and education seriously, and we do everything from development of exhibits to outreach, teacher training, classroom visits, to development of coloring books. There are 2 coloring books in the lower left portion of this slide, the Tiny and Tough Miami Blue Butterfly and Keep Monarchs Safe coloring book, to try to engage a diversity of different audiences where possible. And then we also have different wildlife viewing programs with partners such as the Florida Fish and Wildlife Conservation Commission to get people out connecting with nature and actually looking for these species in their own landscapes. So again, trying to connect people to nature and really talk about the importance of these organisms and the work that we do is really vital. And then just because I like unusual, crazy things, we also have a long-standing collaboration with First Magnitude Brewing in Gainesville to try to use the power of craft beer for conservation. And so over the last 3.5 years, we've launched 11 different beers focused on butterflies to try to raise awareness as well as funding for the conservation effort. So each of these beers that was released, we have a portion of the proceeds goes back into the Florida Museum of Natural History to support butterfly conservation and research. We do public art exhibits, we sell merchandise, we do public educational launch campaigns for each of these beers. And with each of the beers launched, we also try to push the envelope a little bit more to try to ensure new ways of engaging with the public, everything from using augmented reality on the labels, developing videos to go along with this, and even swabbing the butterflies in the field and using the yeast from the butterflies in the brewing process to try to get the butterflies literally in the beer as much as possible. So these have been wonderfully successful, and I've had an absolute blast doing them over the years. And we have a new campaign, which I'm very proud of, which is, again, trying to push the envelope even further. So this is based on the model Black is Beautiful that was launched a few years ago. And so this is a reach out nationally to all craft breweries to try to participate in this campaign, where we provide the beer recipe and the label graphics, and we ask craft breweries to brew this beer, which is called Reign. The campaign is Restore the Reign of the Monarch. And through this effort, we hope 25% of the proceeds generated from the sale of this beer will funnel back into monarch conservation at a national level. And we hope actually to have breweries in Canada, the US, and Mexico participate, because this is ultimately a tri-country migration for this butterfly. And so we partnered up not only with the craft brewing industry, but also with the Xerxes Society for Invertebrate Conservation, who will be our on-the-ground partner for restoring monarch habitat across the nation. And so this beer is scheduled to launch on September 30th of 2022 in the heart of the migration. And so if you have connections or you frequent a local craft brewery, please ask them to be a participant in this campaign. The more the merrier. We want as many craft breweries as possible to sign up. And also, if you're in Gainesville on April 1st, we continue the individual local releases. So this is the latest beer that we're doing on a local level called Tiger Tail Pale Ale, using the Eastern Tiger Swallowtail as kind of the focal organism. So that'll be on April 1st from 4 to 8 p.m. at First Magnitude Brewing, with all the with a portion of the proceeds going to conservation. And we're also doing a tree giveaway for the host plant of this butterfly for the first 100 customers to First Magnitude. And with that, I'll say thank you and happy to answer any questions that you all may have. And I apologize, I did kind of go through this quickly, but I just wanted to kind of talk about the breadth of the work that we do. We also, of course, work on a lot of other insects, but I really want to focus primarily on butterflies for tonight.
Awesome. Thank you so much. This is a really interesting presentation. Any questions? Anyone? Okay, I have a question. I have a question, but also someone in the audience has a question. Someone in the room right now. So I will hand over the mic real quick.
For the Miami blue, you said that the habitat was still mostly intact. What caused the decline then?
That's a great question. So I will say that obviously coastal habitat loss contributed to the decline, but in many areas, the habitat, there's still plenty of what we would consider viable habitat available for that butterfly. So the drivers of decline for that organism are not as clearly understood as for many others. So obviously there is some evidence that invasive species have caused some challenges, including disruption of that ant mutualism. We've had a number of different non-native ants come into Florida over the last 10 years, which are highly predatory, and in areas where we don't see that mutualism particularly well developed, as an example, if the main tendering ant taxa are absent, we see really strong predation in the wild from a lot of different insect species, including those non-native ants. So I do think invasive species has caused a lot of the decline in many areas. We also do believe that mosquito control abatement has caused decline in some areas. Climate change has also potentially affected populations, as has just fragmentation of populations or habitat over time. But there's not like a clear singular factor that this is the dominant driver of decline. So in many ways, we're sort of trying to thread the needle here for recovery without a good understanding of the drivers that initially caused this collapse over the last 30 years.
Okay, thank you.
I had kind of a two-part question about the ants and the symbiotic relationship with the Miami blue, I believe. One was just about oh my gosh. About like whether you do any conservation with the ants or whether the ants are endangered, and then also whether there are any difficulties with like getting funding for conservation with like other insects that aren't as charismatic as butterflies.
Sure. So great majority of the ant taxa that tend this butterfly are quite common. The Florida carpenter ant as an example. And so generally they're not in need of conservation, but we have seen, as I mentioned with the previous question, that in areas where several other non-native ants have come in, we're seeing disruption of those ant populations, the native ant populations in those areas, and potential disruption to the mutualisms that the butterfly needs for or benefits from for survival. So I think there's definitely going to be some long-term continued changes to these systems, but the ants themselves are not in any danger of significant decline for at least the taxa that tend this butterfly. And then regarding the funding question, so obviously, you know, fortuitously, obviously, I do work a lot with butterflies, which are charismatic and generally the most charismatic group for insects. But we do a lot of other work with other insect taxa, particularly native bees or pollinating communities and pollinating insect communities in general. And so it is sometimes challenging getting funding for those. However, we kind of take a different tactic. So we look more at the impact of the community level with those species groups than we do as individual taxa. So it is hard to get individual species or individual taxa funding for a wide range of insects. So trying to look more broadly at how do we look at landscape or community level impacts. And obviously, since insects deliver such important ecosystem services, pollinating insects as a good example, you know, funding from agriculture, funding from grower groups, funding from even pesticide companies can be, you know, available and out there. And we've been also fairly fortunate in getting funding from departments of transportation, energy providers, Duke Energy as a good example, municipalities that might want to develop urban corridors, things like that. So we try to be as creative as possible, but there is actually a lot of funding out there. You just have to think about how it fits the work that you want to do and kind of craft the argument most effectively.
Thank you.
And I will also mention, I forgot to mention at the end, that if anyone is interested in volunteering over the summer or undergraduate research opportunities in my lab, we work on a wide variety of projects across Florida, including several within North Central Florida. So we are essentially always looking for others that might be interested in helping us with that work, especially over the summer.
So, would they be contacting you about that, or is there a website that they can give you?
Yeah, you can contact me directly via my email or through my website. Yeah.
Awesome. Thank you so much. I don't think there are any more questions. So yeah, thank you so much for coming and sharing all your research. Yeah, it was really amazing. So thank you.
Thank you so much for having me.
Have a good night, everyone.